All right, but let me summarize the last lecture. And this is one thing I like to do, where I can summarize in a minute what took an hour and 15 minutes to say. What did we learn, hopefully, last time? We learned that the body wall is overwhelmingly comprised of somatic structures, that the somatic motor structures of the body wall are striated muscle, and that the tendon, the dermis, the ligaments, the bone, the fascia, those are all somatic structures as well, just not motor in nature. That spinal nerves were created for the purpose of innervating the somatic structures of the body wall. The ventral root carries somatic motor axons out from the ventral horn cells out to all of these somatic or striated muscles of the body wall, and the dorsal root is conducting the somatic sensory axons from ultimately the body wall, from these muscles and from the other tissues, back into the spinal cord. That the structure is fundamentally segmented on the basis of the fact that dermamyotomes derive from somites, which were segmental structures, and that has given the body, in some places, a very obvious segmentation, like in the chest, but everywhere segmentation, even if it's not clearly obvious to you. So that is essentially up. And finally, that because of the way the dermamyotomes are divided into epiaxial and hypaxial portions, there is an epiaxial region of the body wall, which is the portion of the body wall composed of muscles that are derived from the epiaxial dermamyotomes and all of the tissues and overlying skin associated with those muscles. And that region is fairly small. It's located dorsal to the vertebrae and dorsal to the beginnings of the ribs. And then there's a much larger hypaxial portion of the body wall composed of all the muscles derived from hypaxial dermamyotomes and all of the tissues surrounding and overlying those muscles. The epiaxial portion of the body wall is innervated by the dorsal ramus of a spinal nerve. The hypaxial portion of the body wall is innervated by a ventral ramus of a spinal nerve. If you know a muscle is epiaxial, by virtue of its position more than anything else, you know that it's innervated by a dorsal ramus. So you're shaking your head. Yes. Give me a name. Give me a name. Name me an epiaxial muscle. The multifidus is a good epiaxial muscle. Very much innervated by a dorsal ramus. Anybody know another epiaxial muscle?
Erector spinae.
Erector spinae, a great epiaxial muscle, innervated by a dorsal ramus. And all the ones we dissected. Not all the ones we dissected on the first day, I should say, because some hypaxial muscles have migrated onto the back and hidden epiaxial from view. The latissimus dorsi is a hypaxial muscle that has migrated from the limb onto the back and hidden the epiaxial muscles from view. The trapezius is a complicated muscle, actually derived from something in the head, which has migrated down from the head and hidden epiaxial muscles from view. And so are the posterior serratus muscles, which we don't care about, and the rhomboids, too, are hypaxial. So there are these hypaxial muscles which make it hard for us to see all of the epiaxials right away in dissection. But when you get rid of those, you can see this column of musculature, dorsal of the vertebral column, and the early parts of the ribs, which are the true epiaxial muscles. And they're all innervated by dorsal rami, and no ventral ramus would ever have the nerve to innervate an epiaxial muscle, and no dorsal ramus would ever innervate a hypaxial muscle. All right, so we have now covered the entire somatic innervation of, or the innervation of all of the somatic structures of the body wall. That's done. We're through with that now. So in order to finish the body wall, we have to consider the innervation of the visceral structures of the body wall. I remind you, you all know this by now, they are the glandular and smooth muscle structures of the body wall, which reside in sweat glands, the muscular walls of arteries and veins, and also the erector pili muscles. They are structures which need visceral innervation. So what we're going to talk about today is the visceral innervation of the body wall structures. And when we've completed, we'll have done the entire body wall. Won't be anything left to do in the body wall. All right, this brings us to, as we know, I hope, the autonomic nervous system. Gee. I just want to lower the lights a little bit, even though that promotes sleep. So if I fall asleep, please wake me up. You know that by definition, the autonomic nervous system is the general visceral motor system, or the visceral motor system, the visceral motor system, the part of the nervous system that innervates smooth muscle, cardiac muscle, and glands. It has as its great distinguishing characteristic from the somatic motor system is that the actual cell body whose axon goes out and impacts or on a smooth muscle or a cardiac muscle or a gland, that cell does not lie in the central nervous system. That makes it very different from the somatic motor system. The somatic motor system cell whose axon goes out to a striated muscle lies in the central nervous system. The ones we've talked about for the trunk, those cell bodies lie in the ventral horn of the spinal cord. When we get up to the head, and you look at all the striated muscles of your head, the cell bodies that innervate those lie in the brain itself. But the cell bodies throughout the body, the cell bodies that innervate smooth muscle, cardiac muscle, and gland don't lie in the central nervous system. That is the overwhelming distinction between it, the visceral motor and the somatic motor system. The autonomic nervous system, the visceral motor system, has two components by named parasympathetic and sympathetic. The sympathetic was the first part to be discovered historically. Sympathies is an old English word for emotions, sympathies, and people realized that when highly emotional states, something was going on, heart rate would go up, your iris would dilate, and things like that. And so the part of the nervous system that was involved in these highly emotional states was given the name of the sympathetic nervous system. Then later on, this other part was discovered, which has an opposite effect generally, and they just named it the parasympathetic system, next to the sympathetic one. Now, when we talk about the visceral motor structures of the body wall, our life is a little made easier by the fact that the parasympathetic system doesn't go to the body wall at all. So all we have to do in trying to understand the innervation of the visceral structures in the body wall is to understand something about the sympathetic nervous system. We'll come back later when we talk about the chest and the abdomen and get our understanding of the parasympathetic portion. The sympathetic nervous system, we're not going to learn everything about it today. We're going to learn about the part of it that controls the body wall visceral structures. And for that, we must return to our friend, the neural crest, because the neural crest is very active, involved in this. So here is now the only difference between this picture and one I showed last time is I made the neural tube and the neural crest gray as opposed to green. You'll maybe see why I did that later, because I use green for another thing later. All right. So otherwise this is exactly the same as a picture we have seen previously. And the neural crest clump, if we're at each, I'm looking for this thing here, at each segmental level, the neural crest clump starts to migrate ventrally, and it splits as it so does, and the two clumps on either side for each segment continue the ventral migration, and one takes up a position in the space between the pedicles of adjacent vertebrae, and that is the spinal or dorsal root ganglion, which medical students saw in dissection. And the other clump takes up a position near the origin of the costal process of a vertebra, and that clump is called a paravertebral ganglion. And that is what's going to hold the cell bodies whose axons go out and innervate body wall visceral structures. So these two ganglia are worlds apart. They're both derived from neural crest, but one is a sensory ganglion, and the other is, in fact, a motor ganglion. So very different. I just show this to give you the position of the paravertebral ganglion at the root of origin of the costal process of a vertebra. Now, this is the same picture as we saw last time, with the exception that neural tube and spinal nerves have been colored gray, all right? But all the hypaxial and epaxial dermamyotomes in pink are there, all the vertebrae and ribs and things like that are there. So it's exactly the same. Now, because it's a very busy picture, I'm getting rid of the dermamyotomes, who can pretend they're going on to do whatever they do. I just don't want them in the picture. And because it's a very big picture, I'm getting rid of the vertebrae. But these things don't disappear in development, you know? They just aren't in this picture because the picture is going to be busy enough as it is. So we're just seeing here the spinal nerves, the little things are dorsal rami, the big things are ventral rami. And someone pointed out, well, you only have dorsal rami for the S5 and CO. That's because there is no hypaxial portion of the body wall when you get that close to the end of your body. At every level, near the ventral ramus, and I'll go back here to point out to you, near the ventral ramus, at every level, is in fact one of these paravertebral ganglia, derived from neural crest. There's one for every body segment. There's one neural crest clump for every body segment. That turns into one dorsal root ganglion on each side and one paravertebral ganglion on each side. For every body segment, there is one of these paravertebral ganglia. They are coloring green, so that's why I changed everything else to gray, so I could color them green. So here we have, you can count them up. For every body segment, you have on the left and right side paravertebral ganglia. If we put that into our familiar picture here, there is the paravertebral ganglion of this particular body segment. And it contains cell bodies derived from the neural crest. And it is also going to be a sympathetic ganglion. It's going to be a part of the sympathetic nervous system. The paravertebral ganglion is going to be a part of the sympathetic nervous system. Therefore, I put in parentheses what is often called a sympathetic ganglion, more often than paravertebral, really, but I like paravertebral. Now, these cells in this paravertebral ganglion are going to have as one of their important roles control of the visceral structures in the body wall for this segment. That means that they have to send out axons to get to the body wall for this segment. But there are only two things going to the body wall, right? The only two things that go to the body wall in any segment level is a ventral ramus and a dorsal ramus. Nothing else gets out there. So if you're going to innervate something in the body wall, you better find a way of getting into the dorsal ramus or the ventral ramus, or you're never going to get there. And so these cells in these paravertebral ganglia during development send out axons which have to either go in the ventral ramus to the hypaxial portion of the body wall for its smooth muscle and sweat glands, or into the dorsal ramus. I don't know where this one's going. Oh, turning into a ventral ramus, and will therefore go out to innervate a sweat gland or an arterial blood vessel or an erector pili muscle in the hypaxial portion of the body wall at this segmental level. Here's another one. Oops, I'm giving it a name, and a very obvious name. This is an axon which came from a cell that lay within a ganglion, the paravertebral ganglion. Therefore, this axon is very obviously called a postganglionic axon. It occurs distal to the ganglion, if you will. Here is another in the body. Sorry, I can't remember how I label or what sequence I do this in. The body itself, because it gives rise to a postganglionic axon, is called a postganglionic cell body. That is a misnomer, but is accepted by everyone. It should really be called a ganglionic cell body. It's a cell body in a ganglion. But because its axon is after the ganglion, it's called a postganglionic cell body. You can't avoid that the way names are given, so that's how it is. So here, yes?
Where is dorsal and ventral spinal cord?
Here, I thought people would have memorized this in their spine, could draw this in their sleep. Here's the gray matter of the spinal cord. Here's the dorsal horn of the spinal cord. Here's the intermedial lateral column of the spinal cord. Here's the ventral horn of the spinal cord. Here is the ventral root. Here's the dorsal root. Here's the dorsal root ganglion. Dorsal ramus, ventral ramus. I just assumed that this looked real familiar to people. Here's another axon growing out from one of these ganglionic cells, another postganglionic axon. It's entering the ventral ramus also. But rather than follow its friend, it is destined to do an artery smooth muscle or a sweat gland or erector pili in the epiaxial region of the body wall. So it has to turn and head backwards in the ventral ramus towards the spinal nerve sensu stricto. And then when it gets there, it can go and turn into the dorsal ramus and get out to the epiaxial region of the body wall. And of course, there are hundreds or thousands of these things. Many more going into the ventral ramus because the hypaxial portion of the body wall is so much bigger, but quite a few going into the dorsal ramus for whatever sweat gland or erector pili or smooth muscle of an artery or vein is in the epaxial portion. This bundle, which contains all of these postganglionic axons going to distribute in dorsal and ventral rami to the body wall, is called a gray ramus communicans. It is called gray because in a fresh specimen, it looks gray because the axons don't have a lot of myelin on them. Myelin is that fatty substance that surrounds many nerve axons for promoting speed of conduction, but these don't have a lot of myelin on them, so they don't look white, they look gray. Yes?
Is there a difference between communicans and communicantes?
Yes, communicans is singular, communicantes is plural. No, that's a good question, because on half the exams, let me just finish that and then I'll get to you. This is called a gray ramus communicans. However, there are many gray rami communicantes in the world. Yes?
Are the postganglionic axons growing after the rami are already formed?
No, the question is, do the postganglionic axons grow after the rami are already formed? And the answer is no. It is the postganglionic axons which create the rami by their growth.
Oh.
The ramus is the name given to all of the postganglionic axons in a nerve bundle you can pick up in your hand with a forceps. That gray ramus only exists because all of these things, hundreds and thousands of these things, are growing towards the ventral ramus to distribute to the body wall. And when they grow, they create the gray ramus by their existence.
And not the gray ramus, but the dorsal and ventral rami. Are they already there before?
Yes, the dorsal and ventral rami have already been created, when this stage of development occurs. Because they were created, why? For the purpose of innervating the somatic structures of the body wall. Right. This is almost like an afterthought. Oh, gee, now we've got some other things we better take care of, too. All right. So that's the gray ramus communicans. Now, look at this. Every single region of the body wall has blood vessels. Most regions have sweat glands, and there might be a few regions that don't, maybe the head of the penis or something like that, head of the clitoris. But they all have blood vessels, and most of them have sweat glands, and quite a few of them have erector pili. But the fact that they all have blood vessels is enough to realize that if there are blood vessels in every part of the body wall, then every single dorsal ramus and every single ventral ramus must be carrying these postganglionic axons. Because if they weren't, they couldn't do anything to control these smooth muscles and glands of the body wall. So there is, in fact, at every single spinal nerve level, a ganglion and a gray ramus communicans. And that gray ramus carries the postganglionic axons to the ventral ramus, where most of them go in the ventral, but a few double back and then go up the dorsal. Have to have one for every segment of the body wall. This makes sense, makes logical sense. And you do. But some of these fuse. So instead of having one nice ganglion for every single spinal nerve, as you did when it all started out, when you were in embryo, some of these ganglia will fuse. Turns out that the first 4 cervical ganglia fuse to form a giant paravertebral ganglion called the superior cervical sympathetic ganglion. That is one that you will definitely see in dissection. The 5th and 6th cervical paravertebral ganglia fuse to form a much smaller ganglion called the middle cervical sympathetic ganglion, which most of you will not see, though some of you may see it because it's pretty small. The 7th and 8th paravertebral ganglia fuse to form the inferior cervical sympathetic ganglion, which can be seen, but most people don't spend the effort to, but it's not so small. Then the thoracic ganglia do not fuse. They stay independent of one another. And then down in the lumbar and in the sacral region, the ganglia do fuse, but they fuse in a sort of random way. Sometimes the 3rd, 4th, and 5th lumbar fuse, sometimes the 2nd and 3rd lumbar fuse, sometimes the 3rd and 5th, 4th and 5th sacral, sometimes the 1st and 2nd sacral. It's just not done in the neat, systematic way that's done up here in the neck. Therefore, you don't actually have specific names for any of these fused ganglia in the lumbar and sacral region. You just know that you're not going to find one ganglion for every single spinal nerve. You might find one that serves, in this case, there's a ganglion that has the gray rami go from that fused ganglion to the two spinal nerves that originally those ganglia innervated. Yeah.
So from person to person, there might be different...
Yes, from person to person, there will be different numbers of lumbar ganglia, and the fusion and sacral ganglia, and the fusion patterns will be different from person to person, and can be different on the left side of a person and on the right side too. But in the cervical region, it's pretty darn constant. So now you have, like the superior cervical ganglia, it was formed of 4 fused paravertebral ganglia. So the superior cervical ganglion has 4 gray rami communicantes going to the first 4 cervical nerves. And the inferior cervical has formed of 2 fused ganglia, has 2 gray rami communicantes. Now the plural comes in more relevant to us. In the majority of people, the IG, that stands for inferior cervical sympathetic ganglion, this one right here. In the majority of people, it fuses, though, with T1 to form a name you may have heard called the stellate ganglion, which is usually pretty easy to find. If you wanted to, in the stellate ganglion, obviously has 3 gray rami communicantes. It was formed of 3 fused ganglia, and it handles T1, C8, and C7 body wall segments. So now we've got all of these ganglia in their more or less adult state. The big question that arises is, you can't just let this be this way and end the story here. Because now the central nervous system has absolutely no way to control what these cells are doing. The central nervous system has to have a way to control when you sweat, when you don't sweat, when your blood vessels dilate and when they constrict, when you get goosebumps and when you don't get goosebumps. It cannot tolerate having all of these ganglia autonomously controlling things the way no one wants to. CNS has to get control over them. And the CNS gets control over them by cells that lie in this intermediolateral column of the spinal cord. And we mentioned that this was the visceral motor gray. This intermediolateral column exists in the spinal cord between, inclusively, the T1 to L2 region. It does not exist in the cervical region. It does not exist below L2. So all of the control of these ganglia is by cells that exist in the intermediolateral gray, intermediolateral column from T1 to L2 of the spinal cord. How do these cells gain control over these ganglion cells? Well, an axon is sent out. Oh, by the way, you see this label is obvious. These are called preganglionic cell bodies. That's very obvious why, because here's the ganglion, and they are proximal, if you will, to the ganglion. They're more centrally located, so they are preganglionic. How do they gain control over these ganglion cells? Well, an axon will grow out from one of them. That axon will enter the ventral root. Now, before we thought the ventral root was just somatic motor. But now we see that between T1 and L2, not only will the ventral root be just somatic motor, it will contain these preganglionic sympathetic axons. So it will also contain visceral motor axons. They grow through the ventral root reach the spinal cord sensu stricto. They grow into the ventral ramus, and for some reason they cross beyond where the gray ramus communicates and then leave. This is pretty standard that they do this. And then they leave and go to the ganglion and synapse on a ganglion cell to gain control over it. And in reality, it probably will branch 10 or 12 times to gain control over 10 or 12 of these cells. So I'm just going to show it gaining control over one, but you must realize that a single preganglionic axon controls a multitude of postganglionic cell bodies. So I've drawn one of these. There's another one I drew that's going to control a different subset of ganglion cell bodies. And of course, there are hundreds or thousands, I don't know how many of these things. And they are less numerous than somatic motor axons. But the way I've drawn it here, it looks like they occupy most of the ventral root, but they are less numerous than somatic motor axons and occupy a smaller percentage of it. They go into the spinal nerve sensu stricto, as I said, into the ventral ramus, and then they leave in a bundle, and their very existence creates this bundle as they're growing, which is called the white ramus communicans, because they are much more highly myelinated. than are the postganglionic axons. So they appear white in a fresh specimen. I must tell you that also, in reality, when you look at a sympathetic ganglion in the chest, which is where you'll mainly look at them, you may find two rami communicantes going to that ganglion, and the one which attaches most distally to the ventral ramus will in fact be the white one. Or you may only see one ramus going to them, because sometimes these are actually all gathered together into a single bundle of nerves. But this is the way it is in cats, pretty strictly, so that's how these all got their names. All right, so now this is pretty darn good, right? Now we've got a way to control all the cells with these ganglia by these cells, and of course these cells get descending input from the brain down through the spinal cord, so now the central nervous system is very happy to gain control over these renegade sympathetic ganglia. And now I've shown you in orange, tiny, but that's, anyway, these white rami communicantes, one extending from every ganglion between T1 and L2, because that's the only place they can exist. Since T1 to L2, parts of the spinal cord are the only parts that have cells, have an intermediolateral column with these cells in them. So only T1 through L2 can conceivably have a white ramus communicans between the paravertebral ganglion and the ventral ramus of T1 through L2. So that's good. We've gained control over these ganglia, but we have no way to control these other ganglia. So what's going to happen? Oh, that's the big problem. The big problem is, how are we going to gain control over the cervical ganglia? How are we going to gain control over the lower lumbar and sacral ganglia? And this is how it happens. Some of these axons traveling in the preganglion, in the white ramus to the first thoracic paravertebral ganglion will in fact not synapse on the cells of that ganglion. Many of those axons will synapse on the cells. But some of the axons will not synapse on the T1 paravertebral ganglia, but will ascend and will synapse on the inferior cervical ganglion. And even if it's fused into a stellate, they don't have to really ascend, because they will just synapse on the inferior cervical part of the stellate ganglion. That's good. Now we have control over the inferior cervical. But some of the axons will pass through the first thoracic ganglion and pass through the inferior cervical ganglion and go up and synapse on cells of the middle cervical ganglion. Now we have control over it. And finally, some of the axons that come out T1, ventral white ramus, will pass through the first ganglion, pass through the inferior cervical, pass through the middle cervical, and go up and synapse on cells in the superior cervical. And in fact, I must tell you, these three ganglia, the superior, middle, and inferior cervical, are a lot bigger than T1, and most of the axons in the T1 white ramus are going to be ascending to higher ganglia. Most of the axons in the T1 white ramus are for control of these higher ganglia, not for control of T1 itself. And similarly, and you must have deduced this by now, that, oh, this you didn't deduce. I thought I was going to go to another. Now, with no logic at all, because these systems are not controlled by logic, some axons from T2 and T3, white rami, will also turn and ascend, just sort of help out. T1 is like too much of a burden on the T1 white ramus to control all of these two ganglia. So some of the axons in the T2 and the T3 white rami also ascend, particularly for the middle and inferior cervical ganglia. T1 pretty much controls the superior. That's a detail you'll learn about later, but I mention it now. So there are all these axons passing through ganglia, ascending to go to higher ganglia to gain control of them. And this is what I thought you might have figured out. Some of the axons in the T12 and L1 and L2 white rami synapse on those ganglia to which they are first brought. But lots of the axons in the T12, L1, and L2 white rami pass through the ganglion to which they are first brought and descend to gain control over the other lumbar and the sacral ganglia. And then finally, which is again something there is no reason to happen, but it just does because the nervous system, sympathetic nervous system is crazy, is that axons that go out, let's say, I don't know what is, if this is 3, 4, 5, let's say the 6th white ramus, and control the sixth ganglion. They shouldn't have to do anything else because the fifth has got control, the seventh has got control. It's as if it's never happy with the way things are. So you find that, that axons, even in the middle thoracic region, don't always synapse on the ganglion to which they are first brought. Some of those axons ascend to a higher thoracic ganglia. Some of those axons descend to a lower thoracic ganglia, even though there's no reason for it to happen, because those lower ganglia have their own white rami going to them. But as a result of all of this ascending and descending preganglionic axons, having passed through the ganglion to which they are first brought, instead of having a collection of sympathetic ganglia along the sides of your vertebrae, you have a chain of sympathetic ganglia. That chain being comprised of the ganglia themselves and all of the axons which are ascending and descending in the chain to find the ganglion in which they are destined to synapse. So you have a sympathetic chain or a paravertebral chain of sympathetic ganglia. Now, let us trace a very simple case of how does a vascular smooth muscle in the sixth intercostal space, that's the sixth thoracic body segment, because the intercostal nerve is simply a ventral ramus of the sixth thoracic nerve, the sixth intercostal nerve. How does it get its innervation? This is something you should be able to do. You start out in the T6 intermediolateral column. The preganglionic axon travels out the T6 ventral root into the T6 spinal nerve sensu stricto, into the T6 ventral ramus. It leaves in the T6 white ramus communicans, is carried to the T6 paravertebral ganglion. In this particular case, it will synapse in that ganglion, and the postganglionic axon will rejoin the T6 ventral ramus by traveling in the T6 gray ramus communicans. That takes it back to the T6 ventral ramus, which is called the sixth intercostal nerve, and that will eventually take it out to some blood vessel that it will synapse on the smooth muscle of and innervate. And that is pretty much, this would be the standard pattern. Now, the only difference was if I was going to be talking about a blood vessel at the fifth cervical level, you would have to have the white ramus enter whatever ganglion I was talking about, and it would have to ascend in the chain to get to the ganglion, which controls the fifth or sixth cervical level. Now, that now covers completely the motor innervation of the body wall. We've covered how the striated muscles get innervated. We've covered now how the glands and smooth muscles get innervated. There's only one thing left to cover before we finish the body wall innervation entirely, and that is visceral sensation from structures in the body wall. And you have a right to ask, what kinds of visceral sensation comes from structures in the body wall? What smooth muscle or glandular structures give rise to visceral sensation? Arteries and veins do. You've heard of people who have phlebitis. That's an inflammatory condition of your veins. That is a painful condition, and that is visceral sensation coming back from an inflamed vein. People can get arteritis, and that is a painful condition, inflammation coming back from deriving from inflammation of the arteries of your body wall. So phlebitis and arteritis are examples, the pains associated with them are examples of a visceral pain in the body wall. I do not have any reason to believe that sweat glands give rise to visceral pain. And I have no reason to believe that erector pili muscles give rise to visceral pain. So essentially, when you talk about visceral pain from the body wall, you're talking about from vasculature. It just so happens, and this makes life so easy for us, that the visceral pain pathways from a visceral or an artery or a vein that is a visceral structure in the body wall follow the reverse of the route that brought sympathetic innervation out to them. So if you have learned the route that brought sympathetic innervation out to that region of the body wall, you know the route that will bring visceral pain back from it, because it's the reverse route. So I'll just finish by hopefully getting a slide that shows you just doing the obvious here, all right? Let's say you have an inflammation of this blood vessel in the sixth intercostal space. it's going to give rise to a visceral pain. And that visceral pain will be transmitted to the spinal cord by a sensory axon, which detects it, not a somatic sensory axon, but a visceral sensory axon associated with a visceral sensory cell. So the pain arises by some chemical stimulus. That visceral sensory axon, click, all right, that visceral sensory axon will travel from the blood vessel, which is inflamed, into the ventral ramus that distributed to that blood vessel. It will run in that ventral ramus back to the gray ramus communicans, and it will travel in the gray ramus communicans to the paravertebral ganglion, and it will travel in the paravertebral ganglion, through the paravertebral ganglion to the white ramus communicans. It will then travel in that white ramus to the ventral ramus, back into the spinal nerve, it just following the exact reverse pathway of the sympathetics out to that blood vessel. Here is the only difference. Once it gets to the spinal nerve sensu stricto, there is no point in it going into the ventral root into the intermediolateral column because there's no cell in there that will understand what's happening. So the axon just deviates and goes into the dorsal root, and its cell body is in the dorsal root ganglion. So the dorsal root ganglion not only has somatic sensory cell bodies, but it has visceral sensory cell bodies. They're of a different size, but otherwise there's no real big difference between them. So this axon has its cell body in the dorsal root ganglion, so we've been tracing the peripheral process of the axon. The central process of the axon will continue in the dorsal root into the dorsal horn, where in fact will synapse on one of those viscerosomatic cells in the dorsal horn. So it's the reverse of the sympathetic pathway, with the exception of going into the dorsal root, where its cell body is located, and then into the dorsal horn. And just to show you this in pictorial form, out here is where our ventral ramus is distributing. The visceral sensory axon will travel backwards in that ventral ramus, loop through the ganglion, the gray and white rami, go up here, cell bodies in the dorsal horn. Central process goes and synapses in the dorsal gray. And that now is, I see what my last change will be. Here was our, you would think I'd look at these slides before I came down here, wouldn't you? Shows you how wrong you are. This is a picture we saw, 5 minutes ago of the sympathetic chain, where I have all of these orange things being preganglionic sympathetic axons traveling up and down the chain to connect the different ganglia. But of course, if visceral sensory fibers are traveling exactly in the reverse route, that means alongside every preganglionic axon, there has to be a visceral sensory axon heading backwards, because they're going to follow the reverse route. So I'm going to change the color of these from bright orange, having used yellow here to be visceral sensory. I'm going to change the color of these from bright orange to pale orange to indicate that wherever you have a bundle which you think is composed of preganglionic sympathetic axons, in fact, that bundle is composed both of preganglionic sympathetic axons and visceral sensory axons from the body wall, which are just heading backwards along the same bundle in the same pathway. Yep, there it went backwards. There it is. Nice, a pale orange color. That's quite important to realize that anything you identify as a preganglionic bundle really has visceral sensory axons running in the same bundle. And that is, in fact, the end of it.